Hello friends, this is Praful and I welcome you back to my YouTube channel Biotechnology. In today's video, we are going to understand another important topic of biology, which is called as biotechnology. So let's start the topic. So what do we mean by biotechnology? Students, when we say biotechnology, there are actually two words which are important here. One is biology and another is technology. And when we combine these two words, it becomes biotechnology. So it is actually a combination of biology and technology. So let me define biotechnology. According to EFP, what is EFP? EFP is European Federation of Biotechnology. Now, European Federation of Biotechnology have defined biotechnology as natural science, cellular products, as well as cellular analogs with the engineering principles. So let me write down. According to European Federation of Biotechnology, biotechnology means integration of natural science, and cell products. Now students, when I say cell products can be certain cell molecules like proteins or nucleic acids or lipids. At the same time, it can be enzymes. So it is integration of natural science and cell products with the engineering principles for the benefit of mankind. So let me repeat, biotechnology is an integration of natural science or cell products with the engineering principles for the benefit of mankind. So let us understand biotechnology. Now, what are the types of biotechnology? What are the different types of biotechnology? Let me tell you, biotechnology can be chiefly classified into two types. One is an old form of biotechnology, or we can say a traditional form of biotechnology, also called as traditional biotechnology. And another is, second one we can say, it is the new form of biotechnology, which is modern biotechnology. Students, we already know about certain food items, like for example yogurt, or for that matter cheese, or paneer, or wines. All these products are involved in the role of microorganisms, and that is chiefly some of the products, like for example curds, they involve the role of like lactobacillus. At the same time, some other products, like for example wine involves the role of fungi, which is yeast. So there are so many different types of products in which the role of microorganisms is very important. And this formation of products by making use of certain bacteria or other organisms is a very old form of biotechnology. I mean, the curds are formed since ages. At the same time, the formation of wine has been through ages. So this type of biotechnology, where we are using the microorganisms for formation of some very important products for the benefit of mankind, is called as traditional biotechnology. So now the question comes is, what is modern biotechnology? So here in modern biotechnology, we talk about genetic engineering instruments. So in modern biotechnology, we understand about genetic engineering. Now, genetic engineering. Now look at the term, students, carefully. Genetic means it involves the genetic material of the organism, and engineering means we are going to make use of certain instruments in order to play with the genetic material of the organism. So we are going to understand genetic engineering. So in students, in genetic engineering, what do we exactly do? Now, as I said, in genetic engineering, we modify the genomes of the organisms. So what are we doing? Here we are modifying the genome of the organism. Now when I say I am modifying the genome of the organism can be a plant cell, it can be an animal cell, it can be a bacteria, it can be a virus, it can be fungi, it can be anything. So basically in genetic engineering, I am modifying the genome of the organism. Now question is, why am I doing it? Why am I interested in modifying the genome of the organism? So the answer to this is, when I change the genome of the organism, students, we all know that genome has the information of the characters of that organism. So if I change the genome of the organism, definitely it will affect the phenotype of the organism. So students, by modifying the genome of organism and changing the phenotype of the organism, friends, we all know the example of BT cotton. So if you understand that in that particular plant, that is BT cotton, it is a resistant variety of cotton which is resistant to the infection of bollworms. Now how has it been made resistant to the bollworms? That is by modifying the genome of the cotton plant. And how did we modify the genome of cotton plant? That was by transferring the gene of another important organism, that was Bacillus thuringiensis. So students, we are going to understand this. So there are basically three basic steps which are followed in genetic engineering. So there are three steps of genetic engineering. The very first step of genetic engineering is, first is, we have to look for a desired gene. The first step is isolation of DNA from the cell. So more often it is the DNA which is removed from the cell, and from the DNA we can get the desired gene. Okay, the desired DNA gene which is obtained from the DNA has to be amplified. So the desired DNA is amplified into many copies. And how do we amplify the DNA segments? That is by use of another important tool, my dear friends, and that is called as vector. So here comes the another important role of vector. So what is vector? So the desired DNA is amplified by using vector. Now what do we mean by a vector? Now students, vector is a foreign DNA molecule. More often this vector, which is often used in genetic engineering, is plasmid. So what is plasmid? Plasmid is an extra chromosomal DNA which is present in the bacteria. So more often this vector which is used is a plasmid. So students, by making use of this vector, we are going to amplify, or we are going to form multiple copies of our desired gene by making use of a vector. And then next what? Now the next important step of genetic engineering is transferring the new plasmid, or we can now call it as a recombinant plasmid, because after transferring the gene into the plasmid is now called as recombinant DNA. Now that recombinant DNA we are going to transfer it into the host cell. So transfer of, the third step is transfer of recombinant DNA into a competent host cell. So what I have done, we are transferring the recombinant DNA into a competent host cell. What is a competent host cell? It is a bacteria. Most often it is a bacteria which is used as a host cell, and very commonly, students, we use E. coli. So E. coli is a very common type of host cell which is used in genetic engineering. So after transferring my recombinant DNA into a competent host cell, what is going to happen? The host cell is going to multiply. And when the host cell multiplies, I am going to get many copies of my desired DNA. Now the question is, what are the different tools we use in genetic engineering? Now students, there is something called as we said, the DNA has to be cut into pieces in order to get the desired DNA gene. And here we talk about some very important tool of genetic engineering, which are nothing but the enzymes. So students, here for cutting the DNA into the pieces, we make use of certain enzymes. So students, there are some special kind of enzymes which are used in genetic engineering. So what are they called? These enzymes are called as nucleases. So students, these enzymes are called as nucleases, and which are of, basically these are of two different types. One are called as exonucleases, and the other ones are called as endonucleases. Now what are exonucleases and what are endonucleases? Exonucleases are those enzymes which can cut the DNA at the terminal ends. We can say the exonucleases are those which cut the DNA at the terminal ends, whereas endonucleases are those which cut the DNA within the DNA, or you can say endonucleases cut the DNA from within. So very common example of endonucleases are, they are called as restriction endonucleases. Restriction endonucleases. So how do they act? As I said, restriction endonucleases cut the DNA. They cut the DNA from within. So if this is my DNA, the restriction endonucleases, they cut the DNA from within. And let me tell you students, this site at which the restriction endonucleases cut the DNA, the site is called as restriction site. So students, it is this particular site, restriction site, at which the particular restriction endonuclease is going to cut the DNA. And so students, in today's video, we understood the basic meaning of biotechnology. So please like the video, share it with your friends, subscribe to the channel BioBuzz, and also put the comments as to which topics you want me to make the videos on. Thanks a lot.